Today I thought I'd share a little bit of our work on tracking and training attention with a cognitive brain-machine interface. I should mention that this is collaboration, at least the first part of this work is in collaboration with Professor Byron Yu at Carnegie Mellon University as part of the CMU ISC Brain Hub grant. I have an interesting connection with Telluride, I should mention, because it looks like many of you all maybe from there. I've at least been there a few times, several times. I was there once as 2009 as a graduate student of Kwabena Bowen, and it was a really memorable experience in many ways. But I'd related a small anecdote. It's the first and only time in my life I've been caught in a cable car probably several tens, if not 100 feet above the ground, where the power or something ran out and the cable cars all stopped and we were all suspended above the ground for about 5 minutes, nerve-wracking experience. And when the power came on, some of the cars moved and the others remained stable. So the car that was behind us actually came towards us, knocked against us, and then our car started moving. So it was it was an experience I can never forget. So I'll never forget Telluride for the rest of my life. So, but for the students here, it's a wonderful place to go, to learn more about collaborative research, really how team science gets done, right? Engineering challenges, etc. And before I start, I'll again, thank all the people who've really done the hard work. There are several students, Abhijit, Suhas, and Siddharth, who worked on the first part of this, this work. And Vishesh and Shivam have worked on the second part of this work. Vishesh is a PhD student here at ISC, and the others are RAs who since moved on to doing their own PhDs. And this work is being ably continued by Dhruv and assisted by Deepak here. So they're also here, so if you want to ask some difficult questions, you can direct it at them. And all my funding resources have all been responsible for a lot of this work. Okay, so let's start with this simple question, what is attention, right? So our brain has to deal with a rich and ever-changing sensory landscape. And one of the classic examples here in this room, for example, is you're hearing me speak, but you're also hearing sounds from the corridor. There may be sounds of somebody moving around, etc. But your brain has to filter out what's irrelevant and filter in what is relevant, right? So that's what we think of as attention. So William James nicely defined attention over 100 years ago. He said, everyone knows what attention is. It is the taking possession by the mind in clear and vivid form of one out of what seems several simultaneously possible objects or trains of thought. And he said it implies withdrawal from some things in order to focus more effectively with others, right? So in order to deal more effectively with others. And so this aspect of attention you will be seeing repeatedly throughout this presentation, this idea of selection by attention. So generally when I begin my talk, I give this kind of a stark demonstration of how limited in capacity our attentional system is. This demo is called change blindness. I'm sure you've seen demos like this before, but this particular image you're unlikely to have seen because we are the ones who developed this particular demo. So what I'm about to share are actually two images. They look very similar. They're going to flash in front of your eyes. So one's going to follow the other. And if you notice the change, right? What's different between the two images, please raise your hand. Don't give it away to the others. Please just raise your hand. Yeah. So they look like very similar images, but there's something hugely different between them. Okay, two. The people who work on attention, I guess, in some way, okay. Not for people who have already seen the demo, right? People who have already seen the demo are not allowed to raise your hands. Okay, three. Okay, not that many. Four, maybe. Okay, so this five. Yeah, so as time progresses, usually there's a sort of a linear curve, right? And people gradually catch on. So what's changing here is this stack of books in this corner. If you notice, that stack of books is appearing and disappearing.
No, that's not what I thought it was. I thought it was the corner is changing.
No, it is that stack of books. So nothing else about the image is changing. So that's fake change blindness. Right? The real one is here, the stack of books. But this is a really stark demonstration of how limited in capacity your visual system is. So when you look around a room like this, you think you can actually see everything at very high resolution. You absolutely can't. The highest resolution is at your fovea, and then it falls off dramatically towards the edges. And attention generally defines what you process with very high resolution, both in the visual and auditory domain.
But wait a minute, you're cheating a little bit, right? Because you have a blank frame in between.
Correct.
If you just switch the frames, you would see it instantly.
Correct. So the difference, though, is that you're supposed to take in the whole frame at once, store it in iconic memory, and then compare it in iconic memory. Because what will happen if I switch the two frames, one after the other, is your movement selective neurons will immediately get activated. I don't want for that to happen, right? So that's the reason why we're putting that little flash in between. You're absolutely right, though. So as a neuroscience researcher, what I would like to do is to understand how attention works in the brain, how attention shapes vision, for example, is an important question that we study. So the visual cortex, so for those of you who are unfamiliar with this picture, this is a side view of the human brain. So that's towards your right is the front of the brain. This is the back of the brain. And your eyes are there towards your right? So the further side. So let me see if this pointer works. Can you all see this? Yeah. So the eyes are here. Your visual cortex is here in the back of the brain. This is the occipital lobe. And this is where all the neurons that process all of that rich information that you saw in the previous slide, the colors, the textures, the shapes, all of that is processed by neurons in the visual cortex. But then there was some area of your brain that was presumably instructing those visual neurons, telling them to process changes rather than aspects of the scene per se. So these areas of the brain, the areas colored in blue, the prefrontal cortex, parietal cortex, in orange, these are all areas that we believe are important for top-down attention control. So what these areas do is they exert control over how visual neurons encode the world and decide what is important at each moment in time for guiding behavior, right? So maybe in this particular demo that you saw, these neurons are, these areas are telling your visual cortex neurons to selectively process changes and to ignore other aspects of the visual scene, right?
So they get inputs from the visual cortex.
That's right. There's actually recurrent loops. There's both inputs from the visual cortex to the prefrontal and also top-down feedback from the prefrontal and posterior parietal to the visual cortex. So this is a very high-level hypothesis of how we believe attention works in the brain. Some of it is valid, but has been validated, but the reason that the biggest sort of motivation that drives our research are disorders of attention. So you may have heard of disorders of attention like ADD, ADHD, attention deficit disorder, hyperactivity disorder, and so on. These are increasingly prevalent in the population, not only in children, but also in older adults. Pharmaceutical treatments like Adderall and Ritalin are the canonical treatments for these types of disorders. Many of you may have heard of these drugs, but I did not know about the pharmacomechanisms of these drugs. I did not know that these drugs actually have the same mechanism of action as certain psychostimulant substances like cocaine and so on. They actually is sort of a milder dose of those types of drugs. So suffice it to say, the long-term effects of these drugs have not been evaluated at all in any rigorous fashion. Many children are being given these drugs over the course of their development, and we don't know what's going to happen, when they reach old age, right? So these are things that make study of attention disorders and how to ameliorate these disorders very important. And what I'm going to propose here is, can we use the brain's own activity as a way to train people to pay better attention without using any of these pharmacological interventions, right? So this is going to be the focus of this talk. So there are two parts to the talk, and I can go into them in as much detail or as little detail as people are interested in hearing. The first part of this talk is already published, so I won't go into it in too much detail. I'm planning to just give you a high-level overview of it. The second part is really about closed-loop neurofeedback training, which is the more hot-of-the-press stuff. So maybe we can spend a little bit more time on that, and maybe also in the future directions. And I should mention that given that this is a informal group, please feel free to stop me at any point and ask questions. I know many of you are here, engineers and electrical engineers and so on. So if you have any questions about any of the background information that's needed, please feel free to stop and ask. Okay. So if you want to do closed-loop neurofeedback, the first thing that you have to be able to do is to measure brain signals. So that's the first order of business. Now, several of the other speakers in the previous days, my colleagues at the Center for Neuroscience, would have told you about techniques that involve invasive brain recording. So where you open up the skull, implant an electrode, and record neural activity directly from the brain. The advantage of those techniques is that they'll give you excellent signal to noise. You can measure spikes, you can measure local field potentials, you can do lots of interesting things. But an average person who wants to improve their attention is not going to give you permission to open up their skull and insert electrodes and all that. So for that, you need a technique that will allow you to record directly from the surface of the scalp outside the head. And this technique may be familiar to many of you. It's called electroencephalography or EEG. So this technique allows you to measure voltages on the surface of the scalp, but how does it work, right? How can you, it looks like magic, how can you measure whatever is happening inside the brain on the surface of the scalp? It turns out that this works because of a fortunate coincidence. The pyramidal neurons, so which are these like triangular shaped neurons here, in the surface of the cortex are all arranged in a columnar fashion. So they have a certain orientation of the processes that both provide inputs into the neurons, which are called dendrites, and also the processes that take inputs out of the neurons, which are called axons. And because of this columnar organization, all the currents sum up when these neurons fire together, when they all generate action potentials together. And when this happens, you can actually just put an electrode on the surface of the scalp, put an electrically conductive medium between the electrode and the scalp, and you can record voltages at the surface of the scalp. So these oscillatory type patterns are typically what are recorded, because when all the neurons fire together, you see an uptick like this. When they all shut down, you see a decrease. And when they fire together, it goes up and goes down and so on. And these types of oscillations you have heard about, alpha oscillations, maybe some of you have heard of gamma oscillations, beta oscillations, theta, and so on. These are all oscillations that can be recorded directly from the electroencephalogram, the EEG signal, from the surface of the scalp. Yeah.
Does that statement mean that the density of synapses at the top is in general higher always compared to the all other lanes?
The top, what statement is this? So I took this from backyard brains, I should say, but which statement are you particularly referring to?
They are very dense with synapses.
Yes, so what happens is, because of the lateral connectivity in the cortex, there's actually a lot of synapses in the upper layers. So that's that exact picture is what you're seeing there. So if I go here, this this tissue that you're seeing here, that's lots of these lateral connections that happen within the cortex. That's how the neurons talk to each other across cortex. And this columnar structure is what allows you to, if the neurons are oriented any which way, then these, that summation wouldn't happen.
But these lateral connections would also be there throughout different layers too, right? It is not like this.
The reason why it's more concentrated above is because all of those dendrites, the inputs, the processes that provide inputs to the neurons, they receive those inputs at those layers, right? So in those layers. So that's the reason they're a little bit more concentrated there. But you're right in that there will be lots of connections also to the cell body, especially inhibitory synapses are typically made to the cell body, not to the dendrites. Okay, so these are, an example of what you would get when you measure physiological oscillations, like innate intrinsic oscillations. But we study a different kind of oscillation, which is called an evoked oscillation, right? So this is called the steady state visually evoked potential. And the way we measure this is by having a participant look at a screen and watch a flickering stimulus. So the stimulus turns on and off. And when the stimulus turns on and off, you actually get an EEG oscillation that is also rhythmic, but not a sinusoid. So you get power at the fundamental frequency, but you also get power at multiples, at harmonics, right, of the fundamental frequency. And this response is called a steady state visually evoked potential. Now, why is the SSVEP of interest to us? Why do we want to flicker stimuli and measure this SSVEP? Remember, this is not a naturally occurring oscillation, right? This is something that we are evoking by flickering stimuli. We want to do this because there's a very interesting result in literature, which has been replicated many, many times, a very robust result by Morgan, Hansen, and Hilliard, and many authors around this time as well. What they showed is if you pay attention, your SSVEP power gets strongly modulated. So in this particular experiment, this participant was presented with two flickering streams, right, of letters here. So this stream was flickering at one frequency, 12 hertz. And this other stream to the right was flickering at a different frequency, 8.6 hertz. And these two streams evoke SSVEP power at the corresponding frequencies, right, 8.6 and 12 hertz. But if the participant starts paying attention to one of those streams, right, the SSVEP power increases substantially in amplitude. The SSVEP power on the other side goes down, and vice versa. So basically, SSVP power changes with attention on average. Was there a question?
Is that related to eye movement? So he's just getting less illumination because he turns one side of the eyes?
So that's an excellent question. Yes, that's an excellent question. Early studies didn't do a great job with controlling for eye movement. So it's possible that some of this effect is actually due to the eye is moving closer to the stimulus itself. But later studies have very strong eye movement control. So, and they have also replicated this result, which suggests that all of this is happening centrally. So the image, the illumination, everything coming into the retina is exactly the same. The modulation of the SSVP power is happening essentially centrally by circuits opening and closing and allowing some information to come in more strongly than others.
Not just a mechanical effect.
No, it is not just a mechanical effect. Yes, correct. It's actually a selection that the brain is doing. Yes. So, given that SSVP power changes with attention, and it is modulated systematically in this way, we ask the converse question, because this has all been shown on average, right? Across many trials, if you average the SSVP power, this is what you get. But can you track momentary fluctuations in SSVP power and use that as a measure for attention, right? This is what we're going to ask. Now, the moment I pose the question like this, it means that I have to have the ability to track SSVP power in a momentary basis, which means I need something like a real-time brain-computer interface. I need to be able to record the EEG in real time, do the processing in real time, and come up with an estimate of the momentary changes in SSVP power. Is this premise clear so far? Because this is going to be the basis for everything we're going to talk about for the next 20, 30 minutes. Okay, so we're going to ask these two questions. Can we track attention with SSVPs in real time? That is the first question. And the second question is, can we train attention with neurofeedback? So the first question is, I'm going to not spend as much if I can, because it's already published. And I'm happy to share the details of the paper with you. There's lots of details that go into designing a real-time cognitive brain-machine interface of this type. And so I'm happy to share the paper with you, and we can discuss details also afterwards if you're interested. Yeah. Okay, so this is more for the students in the audience. So whenever I say brain-machine interface, typically you think of something like this, right? Where we implant a chip into the brain of the human. Typically this may be somebody who's paralyzed, and then you decode information from that brain recording about, for example, intended movement. So the participant wants to move their hand to the right or to the left. You decode that information and then actuate a robotic arm. I think Arindam was maybe presenting something along these lines about moving a cursor with this decoded activity. But what we are going to be looking at is something different. It's a cognitive brain-machine interface. There are two key differences. One is we're not going to record from invasive chips. We're going to record from the EEG. The other thing is we're not going to decode movement. We're going to decode cognitive processes like attention, right? And the cBMI broadly looks like this. There's a presentation system that's presenting something to the participant. The participant's viewing this. EEG data is collected in real time, processed, and then fed back to the neurofeedback system. So the processing typically involves doing something like decoding attention states, and that information is fed back, and then the participant views that updated information, their EEG brain state changes, etc., etc. So it's a closed loop neurofeedback system. So let me present to you a little video here of how this works, and I won't go into it again in too much detail. I'll come back to it again towards the end of the talk. So what's happening here is this participant, again, their face is obscured. They have an EEG cap on their head. They're resting their face on a chin rest. The chin rest also happens to be an eye tracker. So to address that previous question, we are tracking the eyes to make sure they don't move too far away from the central fixation cross. Right? So we're tracking the eyes there. Then what happens is a cue appears which points to one or the other of these two stimuli. And the participant is told to pay attention to wherever the cue is pointing. And if the participant pays sufficient attention, you can notice that that inner disc is rotating and aligns itself with the outer disc. So that alignment is the participant's goal. And the way they're being told they're paying sufficient attention is by tracking SSVP power in real time. So I'll tell you a little bit more about exactly how this task works in a moment. But this is typically how this neurofeedback setup works. Okay, so those of you who are aficionados in the BMI community, the toughest challenge here is bringing down the round-trip latency. So you have to collect the EEG information, process it, and send it back to the participant without too much delay because if you send it back after 500 milliseconds or a second, the participant's attention state would have changed, right? So you have to do it very quickly, and we tried many different things, and for us, certain combinations of hardware-software interfaces worked, and we have a round-trip delay, which is the overhead alone of about 5 milliseconds, which is entirely acceptable for a device of this type. Okay, so now how do we link momentary fluctuations in SSVEP power with attention? So that's the task that I'm about to show you now. So what the participant does is they come into our rig and they're facing the screen, so all of these stimuli are displayed on a screen. So they're fixating here in the center, which means they're keeping their eye right on this fixation cross. And just like the Morgan et al., that experiment in PNAS, two checkerboards come up. In this case, there are two pedestals, which are like these chessboard patterns. They come up and they flicker at different frequencies. One's flickering at 14 hertz, the other one's flickering at 18 hertz. Then a cue comes up, which is that central arrow there in the middle, this one here, as you can see. That arrow points to one of the two pedestals, which tells them where they should be paying attention, right? So this is the one where they should be attending. Then after an uncertain time, and I won't tell you exactly what that time is, I'll clarify that in a moment, these two stimuli appear. These two are standard gratings. So gratings, for those of you who are unfamiliar, are alternating patterns of dark and white, right? So these two patterns appear, and once they appear, the participant has to make a judgment saying whether the grating in the cued side, this target grating, was tilted clockwise or counterclockwise or vertical. So very simple discrimination task. Left or right, clockwise or counterclockwise, and the participant has to make that judgment, but only for this grating, that is the target grating, and they're supposed to ignore the distractor grating, right? And we calculate a measure of accuracy called D prime based on signal detection theory. I won't get into it again in the interest of time, but it's a, you can think of it as a measure of accuracy. I'll be reporting D primes throughout this task. Okay, so now there's another challenge that comes up if you want to, do anything in real time, especially with this EEG signal, which is that the SSVEP is embedded in a background of an incredible amount of physiological noise, right? So these two peaks that you're seeing here are peaks evoked by the flickering stimulus at F1 and F2, that is 14 hertz and 18 hertz. But you can see a lot of other activity happening all through, right? So this is the power spectrum, power as a function of frequency. This huge peak you're seeing here, does anybody know what this peak is? This is the alpha band activity, right? So this is actually, intrinsic oscillations. There are all kinds of other kinds of biophysical oscillations and noise that are happening in the EEG signal. Now, how do you separate out this activity associated with these two flickering gratings from the rest of the background input? For this, we use a technique called denoising source separation, or DSS. This allows us to clean up the signal into these SSVEPs evoked by the two flickering stimuli. So those are the time traces associated with the SSVEPs. Once they've been cleaned up, those are the spectra. And how do we know we are not pulling out just 14 hertz power in the broadband signal? We know that because of how well the SSVEPs are localized. So this is what is called a topo plot, a topographical plot. This is the nose, these are the ears, so you're looking at a top view of the head. And what you see is that when you present a flickering stimulus to one side of the visual field, you actually get the strongest activity on the contralateral side, the side opposite to where you presented the stimulus over the surface of the brain in the occipital cortex. So the stimulus was presented on the left side, your SSVEP shows up on the right side. If your stimulus was presented on the right side, your SSVEP shows up on the left side, right? So this localization, just one moment, this localization gives you a clear idea that you're isolating the SSVEPs, and that too in the appropriate hemisphere. Yep.
So usually when you're paying attention to one particular task, it is often told that the alpha, you see a separation in the alpha. But here I see that the alpha is very prominent.
So the suppression that you see is in the alpha oscillations contralateral to the side where you're paying attention. So here I'm not showing you space-specific alpha oscillations. I'm just showing you a general spectrum of all the electrodes. So alpha decreases contralateral, that is opposite to where you're paying attention, and it might even increase ipsilateral to where you're paying attention, right? So in the hemisphere on the same side as where you're paying attention. So I'm showing you here all the electrodes, right? All the electrodes and all the frequencies. So there's a push-pull effect there also. Okay, so with this DSS, we can get fairly clean SSVEPs. Now, what do we do with all of this work, right? So we projected our EEG signal into this DSS space, and we're tracking it in real time. So we're tracking it moment by moment by moment. We're tracking both the SSVEP on the cued side and the uncued side, right? The 14 hertz side and the 18 hertz side. So we're tracking both of them. Momentarily, you see there's lots of fluctuations happening in the SSVEPs. So now what can we do if we want to correlate these momentary fluctuations with attention? Do a very simple trick. When the SSVEP reaches a certain predetermined threshold, a high threshold, or when it reaches a predetermined low threshold, we trigger the presentation of stimuli. That's it. So I'll explain this in a little bit more detail. So we track SSVEP power in moving windows. Window upon window upon window, the SSVEP power is tracked. When it reaches a certain threshold, in this case, the value 0.7 is some normalized value of SSVEP power, we trigger the presentation of the stimuli. Conversely, on other trials, when it falls below a certain value, another threshold, preset threshold, we trigger the presentation of stimuli, right? So now how does the full task look like? So you have the presentation of the two plaids here, these two checkerboard patterns. They're flickering away. We are continually tracking the SSVEP power coming out of these two patterns in real time. We ask the participant to pay attention to one side, and when the SSVEP power on this side, the side where they are paying attention, reaches a certain high value or reaches a certain low value, the moment we've detected that that has happened, we present these two stimuli. And now the question that we're going to ask, you may have already guessed, is was the participant paying more attention? Were they more accurate when the SSVEP power on the side was, the cued side was high compared to when it was low? So when we cue them for attention, does the SSVEP power on that side momentarily predict whether they will perform better or worse, right?
But isn't it always higher then? Isn't it always higher if they're paying attention?
Only on average. So that's the interesting point, right? So because you can see right here, right? So I'm actually showing you the cued and the uncued SSVEP power on the previous slide. As you can see, they sort of, fluctuate back and forth, right? So it's not always true that the SSVEP power is, on average it is true, but not always. So that's a good question, right? So this is what we're going to ask. And so now, what's the answer? So the simple answer is yes. If the SSVEP power is high, indeed, participants tend to have a higher D prime, a higher accuracy than when SSVEP power is low. And we're sort of operationalizing attention here with this D prime metric. Like I mentioned again, it's an metric of accuracy. So the SSVEP power level, even the momentary level, seems to be a good predictor of participants' accuracy in this task. Right?
This is a tough measurement, right? Because you had to work really hard to get this. I mean, it's real, right? But it's really a tough measurement.
Correct.
I mean, it's barely any difference. Like here in this D prime, right?
Yes, it is. So the estimation statistics suggest that there's actually a clean effect there. So as you can see that, error bar is well displaced off of 0. But the p-value is 0.03, which typically happens if you don't have enough participants, right? So, but the effect is, fairly robust. But I'll show you in a couple of slides an even more robust effect, so maybe that'll be even more convincing. But for those of you who are, in experimental science, these are, fairly robust effects, right? So unlike engineering, experimental, you have to squint a little bit, but, statistics generally tell you the full story. Yeah.
Can you influence the strength by changing, intensity or size of the individual dots or something like that?
Yes, so that's a great question. So can we change the SSVEP power in some way and then, make participants pay more or less attention to those gratings? Those are things that we could have tried, but this was actually a fairly tough experiment to do in terms of the, tracking the SSVEP power in real time. But those are, manipulations of the stimulus we didn't try. We kept both of them at exactly the same luminance, except the frequency was counterbalanced across sides. So sometimes the 14 Hz was on the left and sometimes the 14 Hz was on the right. But other than that, we didn't change the stimuli in any way.
But you wouldn't have to, you wouldn't have to track the SSVEP power. You could just take the behavior. If they are getting better, then they're probably attending better.
You mean like giving feedback based on the behavior itself?
Feedback, just varying the parameters, and see which prerogative gives the best performance.
So you're right. So one alternative way to do this is to try various combinations and post hoc go in and test whether, that could have been done. But we do an estimate on the paper showing that this kind of direct tracking allows you to infer this association with far fewer trials than if you were to do it post hoc, given how noisy the EEG data is. Because here, momentarily, we're actually tracking how the EEG data goes up and down. So we have strong control over these diverged SSVP power states, which you don't always get, if you're doing it post hoc and presenting things at random times, etc. So, okay, so I'll just close off this part by, giving a very quick overview of one more thing that we did. What I told you so far is that SSVP power, when it's high, you have higher retention. When it's low, you have lower retention. But that's a little bit not the complete story, right? Because attention is not really about your alertness or your motivation and things like that. It's really about selection in space. So something has to be high at one location, low at the other, and so on. So that selection component we did not measure in this first experiment. So we redid this experiment again, but this time instead of looking at SSVP power only on one stimulus, we looked at the competitive effect, which is the difference in SSVP power across the hemifields, right? So across the two different stimuli. And then we asked if we play the same game, compute the difference of SSVP powers, and then trigger the presentation of the stimuli based on the difference of SSVP powers between the target and distractor. Do you see similar or stronger behavioral effects? And it turns out that when you do this, you actually see very strong behavioral effects. So when you have a large difference in SSVP power between the two hemifields, that's when you get a large difference in sensitivity. So attention power is higher when the difference of SSVP across the hemispheres is higher, right? So this little trick allowed us to bring in that attentional effect even more strongly and link it to this SSVP signature, right? Okay, so here are some conclusions that we can draw from this portion of the talk. We are able to come up with a novel SSVEP cBMI, which is a cognitive brain-machine interface with short latencies for tracking attention states in real time. SSVEP power states are predictive of higher accuracy. On one side, higher SSVEP power reflects a higher accuracy, but if you actually do a difference of SSVEP powers across the hemispheres, that is across the two different locations, you actually have a much more robust effect on attention behaviors, right? So this is, I'll maybe pause here to take any questions if we're moving on to the last portion of the talk. Anything was unclear or?
It's clear. Good.
Great, yeah.
Yeah, it's very clear, but since you're asking, does it work for all frequencies, stimulation frequencies?
So that's, again, a great question. So we haven't titrated different frequencies, etc. We just counterbalance them across the different sites. And typically, we selected frequencies that were ideal for that particular participant. But more often than not, this combination of 14 and 18 hertz worked out quite well. But they also need to be sufficiently different spectrally so that their powers don't sort of overlap with each other. So we had to also have a little limitation there in terms of how far apart they had to be. Yeah.
Is it compute heavy? Like, is it require a lot of computation to do this real-time analysis? Specialized hardware or anything?
So right now we haven't been using any specialized hardware. It's all been
PC, right?
It's a PC, yeah. Right. So nothing special, but one of the interesting things is, can we do this compute on the edge, right? So I'll come to that towards the end of the talk. Can we do all of these computations, in fact, something more than this, in a device that's integrated with the EEG cap? And actually, we did a very detailed analysis on this, especially for a grant proposal that we were writing together with Chetan. It turns out that at about 16 electrodes you max out. You don't get much more benefits if you go beyond 16 electrodes, and especially because all of this is being recorded from the occipital cortex, you can maybe do this with even fewer electrodes than that.
Just 16 electrodes, so 8 on each side.
8 on each side, that's correct. Yes, so it's not too bad when it comes to the number of channels that you have to process, at least for this signal. There are other signals where we have more channels that we need. Was there a question?
So far I think you are using power as a measure, right? So did you try using any kind of waveforms which would indicate, or does the waveform basically tell you anything apart from when I'm seeing only the power, alpha power, beta power and all?
Correct. So you're talking about maybe things like ERPs, right? Event-related potentials. Those generally take many trials to model accurately. So that's the little bit of a challenge with using ERPs. So that's why many groups studying attention have started using SSVEPs. So they are much more reliably evoked, because when you're flickering a stimulus, you invariably get this strong response to that, periodic response to that stimulus.
And one more question, suppose you are using all this as a setup, right? It's all kind of on some screen or something like artificial kind of setup.
Yes.
Suppose somebody enters into the room, do you see that kind of attention, alpha power when you, it's a change of attention for a person who is sitting here, right? So do you see, did you do those kind of trials, or is it only with the lab setup kind of thing?
So everything I'm about to talk about, I have spoken about and I'm about to talk about is all laboratory constrained. Those experiments that you're talking about are extraordinarily interesting experiments where, I think the field really needs to move in that direction, where people are looking at much more naturalistic settings for attention. But what we do know is that some of those signatures are definitely going to be present there. Like alpha suppression, for example, is very unlikely to be absent in a naturalistic setting. SSVEPs are a different game, because the stimulus has to be flickering, for them to see it, and that's unlikely to happen in that. Maybe unless you're focusing on a tube light or something, I don't know. But it's unlikely to happen in a natural setting, right? So, yeah. So, but those are again, great questions. Okay, so now we'll actually move to the hot of the press results. So these are a little bit preliminary, so please bear with me. So the question here is, can we train attention with neurofeedback? Right? We said, fairly strongly that we can track attention, but can we actually train attention? Can we help somebody pay better attention with neurofeedback? So for this, we use a setup that's very similar to what I said before. There are a few key differences which I'll point out. This is a closed loop neurofeedback setup. So here's your participant. You're recording the EEG signatures in real time using techniques that can allow you to separately measure two SSVEP potentials. We are also tracking their gaze, like I told you. But the key difference here is that instead of presenting stimuli on the two sides, we are going to present two flickering gratings on the same side. So these are annular gratings, annular meaning they're donut shaped. And there's a bar that's embedded in both of them. I'll tell you about the bar in just a moment. But the two gratings are flickering at two different frequencies, F1 and F2. And that's this side where we're training the participant on. I'm going to call this the trained hemifield. On each trial, the participant is going to be cued, right? So this little arrow here, it comes up and it tells the participant, now pay attention to this grating. If the arrow is pointing upwards, they have to pay attention to the upper grating. If it's pointing downwards, they have to pay attention to the lower grating. And the way they get neurofeedback, again, based on SSVEP power, is based on the difference of SSVEP powers like we discussed before, F1 minus F2. So if they're paying better attention to the target grating, this will be a large value. If they're paying worse attention to the target grating, this value will decrease, right? So this is the basic neurofeedback setup. Okay. So I'll show you a small video of what's happening before I explain what that bar does, right? So here's the little arrow in the middle saying pay attention to the upper grating. And now there are two gratings. They're flickering at different frequencies. The reason you see some strange beats here is because interaction between the phone camera and the screen camera. So they're actually rhythmically flickering, right? So now what's happening with this bar in the middle? So let me restart this demo. So what's happening is that if the participant pays better attention to the upper grating, there's a bead, right, that's sliding down the bar. So that bead will continue to fall as long as the bar remains vertical. But if the participants paying better attention, the bar will become horizontal and the bead will stop falling. So the participant's goal is to prevent that bead from falling off the bar. So if you pay attention quickly and you pay sufficient attention and you pay sustained attention, the bar will become horizontal quickly and the bead will stay put on the bar because it's not going to slide off anymore. But like in this trial where the participant was not able to pay enough attention to the above grating, the bead continues to slide because the bar remains close to vertical. It keeps sliding and at some point it will slide off and the trial is over. Right? The participant has failed the trial. And this is happening, just a moment, this is happening, much faster than this. I've slowed this down like 4x to present to you, all of these different dynamics. But this creates an urgency for the participant to pay attention quickly and in a sustained manner at the location of neurofeedback in order to enhance the SSVEP power at that location. Yes?
And I may have missed that. Is paying attention defined as having the larger SSVEP?
Correct. So it's operationally defined based on our previous study that the better attention they pay, the more SSVEP power difference there will be between the two gratings. That's correct. Yes. Okay, so again, just to orient you to the task starts, the 2 flickering grating is up here, and the goal of the participant is to get the bar horizontal before the yellow bead falls off. Because they don't pay enough attention, they don't get their SSVEP power up in time, or if they don't keep it up for a long time, the bar is going to go back to vertical again and the bead is going to slide off, right? This introduces a strong urgency signal because the participant has to do this quickly. And one of the reasons we believe we are seeing robust results is because, there are many BMI, protocols out there in literature, but very often they provide feedback in the form of audio or in the form of some bar that's changing in height and so on. And while some participants may be genuinely motivated to do these tasks, there are others for whom that may not be something as interesting to do, right? They may not necessarily want to keep that bar high or, change the pitch of the tone to whatever desired level the experimenter wants. But this type of manipulation, where you're gamifying the interface, makes the participants go berserk, right? Because if they're not getting that, stick horizontal, the bead falls off and then they feel like they've lost something. And so they go back and then they do it until they actually get this task right. And so we were able to essentially train all of our participants well with this task design. I should mention also that the way we measure whether the neurofeedback worked or not is using an independent task. It's called a continuous performance search task, very similar to a standard search task. The only difference here is that you have a little target that appears in the middle, in this case a black square, and then the participants are cued to attend to one of the 4 quadrants, and these arrays of shapes keep flashing on and off. I'll show you a video of this also in a moment. And whenever the participant sees the target appearing in the quadrant, in the appropriate cued quadrant, they have to push a button. And if they don't see the target appearing in the cued quadrant, they don't push any buttons. They refrain from pushing a button, right? So the goal is to find the target at the cued location. And this can happen either in the trained or the untrained hemifield. Remember, I told you that each participant is always trained on one side. So that's the trained hemifield. But this cueing can be done either in the trained or the untrained hemifield, right? So let me just give you again an example of what's happening here. So in this case, the black circle was the target. And the participant is now cued to attend to the upper left. So you can see that whenever this black circle came up there, they had to push a button, right? Otherwise, you should not push a button. As long as the target circle does not appear, or as long as, there's nothing else happening anywhere else, you should not push a button. When you see the target shape, you push a button, right? So this is a CPSC task. Okay, so now the way we structure the whole experiment, yeah.
Is it just the shape that has to match or also the color?
Color, both. Shape, color, both. Yeah. So the target itself has to match exactly. Right? So size, everything. Right? It has to be exactly the same. So just to avoid confusing participants, we don't present things of dissimilar sizes. But we did try that, but it was too confusing. Right? So what we do is we have one session of the CPST task. This is a baseline task, followed by the neurofeedback training. That is with this flickering, annular gratings, followed by another session of the CPST task. And the goal is to compare performance in this baseline task, the CPST task, before and after neurofeedback training. So our goal is to see if neurofeedback improves attention, right, in this CPST paradigm. Okay, so before I tell you whether it does or not, the first question here is whether participants are able to modulate their SSVEP power with this kind of neurofeedback training. And that's the evidence I'm showing you here. On the y-axis here is a normalized measure of SSVEP power. On the x-axis here is time relative to the trial end. In this plot, participants are paying attention to the F1 grating and ignoring the F2 grating. And you can see that there's a gradual increasing linear trend. There's an average across 8 participants when they're paying attention to the F1 grating compared to the F2 grating. Right, so in the F2 grating, the power is actually reducing. And the reason it's reducing, remember, is because we're taking a difference of SSVEP powers as the source of the feedback. So the larger they make this difference, the more stronger the feedback is, right? So they're actually increasing that difference as much as possible. In some sense, the opposite pattern is happening when they're paying attention to the F2 grating. There are some baseline differences in SSVEP power between these two, but even then, they're able to continuously increase the power of the F2 grating when they're asked to pay attention to the F2 grating. And there's a trend, a decrease of SSVEP power in the F1 grating, right? Now, this happens for the actual neurofeedback participants. That is, we have 8 participants in this study. And another 8 participants were tested with a yoked group. That's a control group. And what we do for the yoked participants is there are many, many different ways to do controls in neurofeedback design. So one is not to give them neurofeedback. The other is to give them random neurofeedback and so on. But each has its advantages and disadvantages. The thing that we did is to take the EEG activity from one actual neurofeedback participant and pretend that that was actually driving the neurofeedback for the yoked neurofeedback participants. So actually each of them is synchronized to one of the other actual neurofeedback participants, and they're basically seeing the same kinds of dynamics that the actual neurofeedback group would have seen, except that their brain activity is not controlling anything on the screen, right? So in these participants, we actually don't see these kinds of modulations of SSVEP power. The modulation is clearly apparent only in the actual neurofeedback participants, right? So this is the evidence that the brain state is modulating with neurofeedback training. Okay, but does it improve attention? Do you see differences in D prime, right? So that is the million-dollar question. And what we see in the trained hemifield is indeed statistical evidence for that. The actual neurofeedback group, those are all the orange dot guys here. The x-axis is the performance before neurofeedback, and the y-axis is the performance after neurofeedback. And if there was no change, the points should be scattered around this diagonal line. But what you see is systematically points being above the diagonal line, suggesting that after neurofeedback, D prime has increased, right, has improved. For the yoked neurofeedback group, not as much. It's not a statistically significant improvement. On average, they all seem to be no different from baseline, right? What is also interesting is an unexpected result, is that this happens also in the untrained hemifield. So again, remember that all the participants were being trained with these flickering gratings only on one side and not the other. But if you test them on the side that they were not trained also, that is the untrained hemifield, again we see a statistically significant improvement in their performance, suggesting that for whatever reason, whatever mechanism we don't fully understand yet, this neurofeedback is generalizing across space. It's actually giving them attentional benefits that extend across to novel locations that were not trained specifically for the neurofeedback. Yeah.
Seems to be stronger.
Potentially even stronger, right? So that's actually a bit surprising, but I think that scatter in the data, I mean, I'm not convinced that it's a statistically significant difference in the effect sizes. So that's something that definitely can be tested. Okay, so now do we have actually any evidence in the brain as a basis for this kind of improvement in attention abilities? So for this, what we did is we took the broadband spectrum and we tried to decode where the participant was attending. In the CPST task, we tried to decode their attentional locus with a linear decoder based on EEG spectral properties. And if you're interested, I'm happy to talk to you more about this, but it just looks at the spectrum and looks at features drawn from the spectrum. And since there are 1 of 4 quadrants that participants can be attending in the CPST task, we just look for which of the 4 quadrants. Can we decode which quadrant they're attending on each trial in the CPST task? And what happens is, for the neurofeedback participants, decoding accuracy improves following neurofeedback. So what that suggests is that the brain states which correspond to where they're paying attention become more separable across the between and before and after the neurofeedback session for the actual neurofeedback participants compared to the control participants, right? So this is some neural basis also for why we believe these effects are happening. There are many, many more analyses here, but I'm not going to go into them in the interest of time, but I'm happy to chat with you afterwards, like controls and so on that we've done. Okay, so just to conclude this portion of the talk, So we're presenting a novel neurofeedback design, including this CPST, which is a baseline task. This incorporates an urgency signal, and we strongly believe that the reason why this is working as well as I've shown you is because of this urgency signal. Very often, BMI tasks just have some bar or some kind of tone that plays that participants may or may not be particularly interested in terms of modulating their brain states. But providing this kind of gamification and this kind of urgency signal makes participants really engaged in the task. Right? So I believe that's one reason why this is working. Brain activity seems to improve attention both in the trained and untrained hemispheres. At least within the session, you actually see these improvements in attention. And accuracy for decoding the attentional locus based on the EEG signal also improves following neurofeedback. So there's some mechanistic basis also for that behavioral result. Right? Okay, so what are some of the future directions? So I'll just conclude with these. So one of the first and most interesting directions is a collaboration across ISC. Some of you may have already heard of this. The goal is to build here a neuromorphic coprocessor. So you now have a paradigm that's going to allow you to record EEG signals in real time and train somebody's attention. So can you do something with that on the edge, right? So can you put a chip that has the ability to take these attention signals, produce this low dimensional output, which can potentially be projected even onto a VR display, right? And then you have this self-contained setup that the participant puts on, trains themselves for 30 minutes every day, and then can we test if over time their attention abilities improve? So we are trying to now build this together as a collaborative effort. Several labs are involved in this. Chetan's lab is involved, Hardik's lab is involved. Many of us are coming to Arjun from IIT Kanpur. We're all trying to come together and make this happen. This is called the Moonshot Project because we believe it has like limited chances of working, but if it works, we're going to be very happy, and it's something that's going to be a useful tool for many even patients following stroke and so on to be able to use these kinds of devices for cognitive neurorehabilitation. Right? Okay, so this is one type of project. The other type of thing that you can do is actually play games with it. So just in the interest of time, I'll actually show you one little thing that we have done in the lab. And that is to try to control a little rover that's here using this EEG neurofeedback signals in real time. Right? So this is a very standard little robot. The only key thing here is there's a little mobile phone seated on the robot, which is giving you real-time information about where the robot is, what it's seeing ahead of it. And the goal is to navigate this robot with this SSVEP signal, right? So I'm going to show you that in the next slide. So here's the robot that's sitting on the floor. And this is what this camera from the mobile phone is being streamed in real time to the person who's viewing that display. But in addition, that person is getting 3 flickering lights here that evoke SSVEPs at 3 different frequencies, right? And so when they pay attention to this light, the bot moves forward. When they pay attention to this light, the bot moves left and right and so on, right? And they, keep their attention centrally, the bot stops, right? So the engineering challenge here is to do all this in real time, make sure the camera display is streamed to the participant without too much lag, decode the EEG signal in real time, then navigate the robot and make sure it doesn't hit any walls. So this is one of the successful runs where it actually didn't hit any walls. So you'll actually see that he's almost about to hit, right? So, but then just in time the decoder worked, and with all its non-stationarity, it turned the bot around, and now we're able to come back to home base. And remember that this is all happening on the other side of the participants. There's a partition here, and the person who's actually navigating the robot cannot see it directly at all. All he's getting is this video feed coming from that mobile phone that's sitting on the bot, right? So we can play some interesting games like this. And with that, I'd like to end with a shameless plug for the lab. We call ourselves the Cognition, Computation, and Behavior Lab, and we use a range of different techniques in the service of understanding how attention works in the brain, including neuroimaging techniques and techniques that allow us to measure activity and connectivity in the brain. There's a slightly older photograph of the group. We're all red-faced and exhausted after open day, which is a day that we spend explaining the brain to public who come into ISC who want to learn more about the brain. So we do this under the sun. So we receive all the March sun in Bangalore. And so the reason we are looking a bit tired is because it's the end of open day. And with that, I'd like to thank you for your attention. And